So, hi again, everyone. I'm happy to be back and happy to announce our next speaker of this second session. I'm happy to present the CEO of Asceneuron, based in Lausanne, Professor Dirk Beher, and he will talk about OGA inhibitors to target intracellular protein aggregation and neuronal dysfunction in Alzheimer's disease and Parkinson disease. Please go on.
Okay, thank you, Emanuele, for this introduction. I'm just gonna bring this to full screen mode, which should be now the case. So just to start this presentation, I happen to be the founder of the company and also a shareholder, hence here are my disclosures. We can skip over quickly. So I think what we are really interested in is proteonopathies, and these are diseases which are characterized by the intracellular aggregation of proteins. And the key interest here is either tau-related disease, where you have tau protein, or synucleinopathies, where you have alpha-synuclein. And I think what we like here is the parallels, because we know the genes. In the case of tau and synuclein, we know the pathology pretty well. And what is important is that mutations in the genes cause disease. So mutations in tau cause frontotemporal lobe dementia, some rare disease, also mutations, progressive supranuclear palsy. And on the other hand, as you know, mutations in synuclein, alpha-synuclein cause Parkinson's disease. And also there's a gene dosage effect with a gene triplication that also is sufficient to cause disease. It's pretty much similar to what you see in Down syndrome for Alzheimer's disease, where you have a triplication of chromosome 21, which carries the APP gene as well. So also, it's fair to say that we're looking here at the large number of diseases. When you look at tau, you know, the most prevalent one is Alzheimer's disease, but there are also primary tauopathies where you only find tau as the disease-causing pathology. Parkinson's disease, the same thing. You know, you have Lewy body dementia and multiple system atrophy. And I think what we like the most is what you see on the left is that we have tools in Alzheimer's to really quantify the formation of these insoluble aggregates, as you can see on the tau PET on the left-hand side, where it can differentiate Alzheimer's and healthy controls. And what you see is that there's a very strong correlation between clinical decline, brain atrophy, and the tau accumulation, and that's also substantiated. Aducanumab is quite discussed controversially these days, but when you look at other antibodies that clear beta amyloid, whenever you see some signs of potential efficacy, you also see changes in tau. So it tells us that these the aggregates are actually causally related to disease, and we share that view as well. So the question is, how can you target this? And I think what we're targeting is the O GlcNAc cycling. O GlcNAcylation is a specific post-translational modification of intracellular proteins. So it's the addition of single N-acetylglucosamine units to serine or threonine residues of proteins in the cytosol. And this is done by a unique enzyme called O GlcNAc transferase, or OGT. When you knock out OGT and you lower the levels of O GlcNAcylation, you get neurodegeneration in the brain. So this is very important that the intracellular proteins are always glycosylated. And what you see in disease is a drop of these levels. This has been demonstrated for Alzheimer's disease quite convincingly, and it's also seen in epileptic brain and other diseases. And I think the question is, how can we rebalance the system here? And one way to do that is by blocking what we think is the eliminating pathway. So that's the OGA enzyme, which actually clips off these modifications. And it's also good for drug discovery because enzyme inhibitors are a modality that has led to successful drug approvals. I think you may ask, you know, why is this really important? I think it's very important because a number of people, and this is not only us, you know, this is big pharma companies, have actually shown that when you block O GlcNAcase, you increase the glycosylation of the tau protein, and thereby you prevent the formation of neurofibrillary tangles. But there's also data on alpha-synuclein that in vitro you can actually prevent the aggregation of alpha-synuclein. And there's also recent emerging evidence on TDP-43, which is also involved in frontotemporal lobe dementias and ALS. So essentially, you have a number of proteins, they tend to form these intracellular aggregates, and it's beneficial to promote the glycosylation. Besides, there's data on synaptic function, so you can decrease aberrant synaptic signaling, so in this case, reduction of hyperexcitability. I think we also have seen literature reports on neuroinflammation osteoglyosis, and I think I show something here on the left-hand side, which is some of our own data in the Parkinson's mouse model, where you see the vehicle-treated mice, they have substantial osteoglyosis after 6 months. And on the right panel there, you see a treatment with one of our OGA inhibitors where you see hardly any osteoglyosis seen here by GFAP immunoreactivity. It also prevents, these compounds can prevent age-related cognitive decline. And we have also data you will see later in these transgenic mouse models improve survival. Just to briefly share a little bit what we are doing. So we have 2 compounds in development. One, which we call ASN12290 or ASN90, is a phase 2/3 ready molecule, which we're developing for the orphan tauopathy PSP, progressive supranuclear palsy. It's actually a motor disease where you have midbrain accumulation of tau, and exclusively tau, so there is no other comorbidity or copathology seen in PSP. We have another molecule, ASN121-151. You will see some of the data here as well, which we call ASN51 for the short form, which has completed a phase 1A study, which we're developing for Alzheimer's and Parkinson's disease, mainly because it's a once-a-day drug, a low-dose drug. You will see some of the data later. I think it's very important to mention that these inhibitors, and this is not only ours, also from our competitors, actually reach therapeutically relevant exposures in human plasma and also in human CSF. As you may know, CSF is always used as a surrogate for free drug in the brain. They are seen to be safe and tolerated both in GLP toxicology studies and also initial human studies, which is important because this OGA enzyme is actually ubiquitously expressed and has thousands of substrates. But I think we are trying to lower lipids in case of diseases, or if we're trying to change the insulin levels, increase them, and for diabetics, what we're doing here is we're just resetting the system to get back into, when you remember the slide before, into the green zone where we have the optimal zone of glycosylation. We don't want to do anything too dramatic, and this is probably the reason why these compounds are relatively well tolerated in a number of these studies. I'm just going to run through some of our data. So when you do tau experiments in vivo, what you need is mice that overexpress human tau carrying mutations that are found in frontotemporal lobe dementia. And on the left-hand side, you see a mouse model. That's one from Michel Goedert, which is the P301S, a mutant of tau. It's a rapidly progressing model, and you see on the top left panel, you see tangles forming. This is Galea silver stain after a certain period of time. So this is 3.5 months of vehicle treatment. So these mice are about 5.5 months old. And on the bottom, you see the treatment with a compound, in this case, ASN12290. You see hardly any neurofibrillary tangles forming. That's in line with what we know from the literature. In the middle, we also switched to another mouse model, the P301L mice from Fred van Leuven's group. And what we see here is a clear survival effect. You can appreciate the differentiation between the vehicle and the other colored lines here on the top and the middle top panel. We see an effect on motor efficacy. The clasping score here is a bad thing, so it's a disability score. So you want to push this down. And then on the right-hand side, we see something that's actually quite intriguing as well, which is an effect on lung function. So these P301L mice have a problem with breathing. This is well documented in literature, may have to do with the brainstem atrophy or brainstem tau. And within a 4-day treatment, we see an improvement. It's remarkable because this has nothing to do with neurofibrillary tangles. These tangles take months to build up, whereas the lung function can be recovered. This can be treated within 4 days. So this has, to our view, more to do with tau biology and the biology of this abnormal tau in these mice. What we also see are some nice pharmacodynamic data. So you can actually measure the organocylation of tau with a specific antibody, recognizing the serine 400 when it's organocylating. You can see on the left panel, when you dose the drug to steady state and then you do a time course, you see nice increase after the last dose, after 8 hours in this O-tau. And remarkably, you also see an increase in soluble phospho-tau, in this case, serine 356. We don't overinterpret this, but it tells us that the treatment with the OGA inhibitor has an immediate effect on tau biology. So it's changing phosphorylation, and we can also explore this as a biomarker in the clinic. So going forward, as I mentioned at the beginning, there's also data out there showing that alpha-synuclein is a substrate and it prevents organochelation, prevents aggregation. We treated a Parkinson's disease model overexpressing wild-type alpha-synuclein. And what you can see here in the motor function assay that at baseline, you can already see an inkling that these line 61 mice have a different profile compared to the wild-type, which are the gray bars. And when you look at 12 weeks and 24 weeks of treatment, you see that there's a clear treatment effect for the compound, which is the first time that people have shown efficacy in a Parkinson's model. It's very important to mention that we did the same experiment with another inhibitor, which is the ASN51, and we get the same results. You know, essentially, we did only 12-week treatment, and we again see a quite substantiated treatment effect in the beam walk, you know, where the number of slips are counted. And this is giving us the confidence that the previous data is not just a fluke. These are highly reproducible data, and in this case, this compound has actually a better PK, so it works already at 3 milligram per kg, whereas the other one before you needed a higher dose. So this gives us a really good opportunity to develop these drugs for tau and alpha-synuclein related diseases, because we're seeing efficacy in both tau and alpha-synuclein models. Just to give you a snapshot about the human situation, so this is this compound ASN51, where we did a PET study in human healthy volunteers. So what you see on the left-hand panel is really high binding of the PET tracer to the OGA enzyme in the brain. So it's highly expressed in the brain. And what you do see is competition if you do a pre-dose with ASN51, and you either scan 6 or 48 hours after the dose, so you give the tracer 46 or 48 hours after giving the oral drug, and you see nice displacement. So you see the reduction in the labeling. And you can actually plot the PK, which is the gray dots here on the middle graph, and then you can calculate the brain occupancy. And what you see is that even after 72 hours, you still have over 85% of the enzyme blocked by ASN51, which is probably the longest half-life in this class. So this will mean we have here a low dose once-a-day drug we can develop, and the anticipated dose is around 5 to 10 milligram. I think another experiment we did is shown here on the right-hand side. We actually showed also that in human PBMCs in these volunteers, there's an increase of the glycosylations, which is the result of blocking the OGA enzyme and the OGT enzyme still being active. And the relevance here is quite high, because when we look in the mice, when we look in PBMCs in the peripheral blood mononuclear cells on the top left, you see an increase with the compound. And this is also mirrored by the increase on the bottom and the change of the tau glycosylation. So, and you see in the middle panel, the correlation here, it's quite high. So that means what we're seeing here in the peripheral mononuclear blood cells is a reflection what will happen in the brains of these healthy volunteers. It's quite exciting data here to have. I think just to summarize here, I think there's multiple inhibitors in the clinic, and these compounds prevent the elimination of organochelation, and hence they're protective against neurodegeneration. We have a rich target biology here and a multimodal mechanism of action, so we can actually target tau and alpha-synuclein pathology in parallel. And as you know, this is what's happening in Alzheimer's disease and other diseases. The LAG2 carriers of LAG2 mutation carriers of Parkinson's disease also have extensive tau pathology. I'd like to thank the team here at Asceneuron. There was a huge group effort, both preclinically and clinically, to get 2 molecules to the development stage. I'd like to thank our advisors, collaborators, and funding sources going forward here as well. And I'd like to thank you for your attention.
Thank you very much for your talk, Dirk. It was very interesting. I think that we can just move to the last part, so where we can have a panel discussion where everyone can ask questions. Okay?
Okay, thank you, Emanuele.
See you.